Hello, everyone. This is now day 2 of the lectures that I'm intending primarily for my physics class, but I'm trying to also get a sense of how Zoom works and how I can create content that will be useful to that physics class. So when you see this video, please feel free to comment and share, and I would be very curious to see what can be done to make my teaching more effective. The lecture topic I'm going to discuss today is of contemporary concern, or of particularly timely concern, and that is how those COVID tests or how those coronavirus tests actually work. And I'm talking about specifically the types of tests that are based on a technique called reverse transcription polymerase chain reaction. So let's go ahead and get right into it. Go ahead and share the screen here. Already had a few bumps today. This is actually the third time I'm trying to record this, so yeah, please be patient. All right, so we're going to discuss how coronavirus testing actually works. And if any of you are wondering, I got my PhD in synthetic and cellular biology at Caltech. So this is a technique that I have actually used in research before. So I'm not a random person on the internet who doesn't know what he's talking about, of which we've seen quite a few. Anyhow, before we start talking about how the virus testing actually works, we need to establish a few basics and fundamentals. And one sort of good place to start is with this thing called the central dogma of biology. And this is something that you see again and again and again in biology courses, and it's kind of easy to forget if you haven't taken biology recently. The basic idea behind the central dogma of biology is DNA, which is the genetic instructions for almost every organism on Earth, I believe every organism on Earth, is transcribed into a messenger, something called ribonucleic acid, or RNA, which is only single-stranded. And that RNA is further translated by something called a ribosome in both prokaryotes and eukaryotes, and that is what ultimately makes up a protein. For the purposes of this lecture, we will say that a protein is what a cell is, or what a cell does. This is a gross oversimplification. There are a lot of other important macromolecules in the proper functioning of a cell, things such as sugars, carbohydrates, fats, and RNA actually has functions beyond just carrying a message from the DNA to the ribosome. But for the purposes of this lecture, what we're going to say is that a protein, or what proteins a cell is making, is pretty much defining that cell's function. And what that cell is doing. Again, a gross oversimplification. So other biologists out there, yes, I do say that is an oversimplification. A way of thinking about this is that the DNA is like the instruction manual for the cell or for the entire organism, and the RNA is an individual set of instructions or a message. So only part of the DNA at any one time is actually being transcribed. Also, a single copy of a gene on DNA may be used to make many thousands of copies of RNA, again, depending on what the cell is doing and what it needs at that particular point in time. Now, viruses are capable of hijacking this process at two particularly critical points. So first off, we have to remember that viruses cannot reproduce by themselves. A virus that's just sitting on a surface or in a droplet is, in a sense, kind of inactive. It's just sort of there. And it's not able to make copies of itself without the use of some kind of machinery, some kind of cellular machinery, which it is hijacking. So the way the virus reproduces is to hijack this process in some sort of a host cell in order to make viral proteins and make viral nucleic acids. A virus is basically just a nucleic acid that is in some kind of a protein coat. So it has to make copies of that and also force the cell essentially to package that virus, which can then be exported, often with deleterious effects to the host cell. The way that viruses do this is either by injecting DNA into the host cells or injecting RNA. So a DNA virus is inserting itself here. You have a foreign piece of DNA that is coming in. It is integrated into the chromosome of the host cell, and then that DNA is then copied into RNA, or rather transcribed into RNA, and that is then used to make the viral proteins. Alternately, some viruses inject RNA instead, and that is used directly by the ribosomes, or the ribosomes are tricked into using it to make, again, more copies of the protein and that is necessary for the virus to package itself, as well as making more copies of the ribonucleic acid that in essence is the virus. There is one specific class of viruses that actually will inject RNA, and then it will have a reverse transcriptase. So in other words, there are some viral proteins that are capable of taking RNA, turning it into DNA, and then integrating that DNA into the host DNA or the host genome. This is something known as a retrovirus, the most famous example of which is HIV. So the coronavirus or the coronavirus family is hijacking the process right here in the RNA stage. It is injecting RNA, which is then used by the host ribosomes, in this case the human ribosomes, to make more copies of the virus. And that's kind of shown here. We have here a coronavirus. It's not COVID-19, but it's of a family of that particular virus class. The RNA gets injected, and the ribosome, this is a native ribosome of this native cell, is being used to create a virus polymerase. So that's sort of one of the first things that ends up getting transcribed. And this virus polymerase starts making a whole bunch of RNA. It starts making copies of all of the RNA that was inside of this protein coat. And this RNA is then acted upon by ribosomes within the host cell to make all kinds of things. It's used to make, again, the outer casing of the virus. It's used to make the adapter proteins that allow the virus to attach itself and infect other host cells. And it's used to basically package together this entire virus, which can then be exported into the extracellular space. So what testing is aiming to do is to find things that really shouldn't be there inside of a human. So let's say that we have a young woman here, we'll call her Stacy for the purposes of this video. And Stacy could either have the coronavirus or not. But let's say that she's asymptomatic. In other words, she is not feeling sick, she's not looking sick, she feels perfectly fine, but she could very well have the virus inside of her. If she's healthy, what we're expecting to find inside of Stacy is normal human cells, things like cheek cells, things like red blood cells, things like liver cells. In this particular case, we'd be concerned about, cheek cells or things in the respiratory tract. However, if Stacy is infected, we'll not only see the things that we are expecting to see, we're also going to see particles of COVID. Just kind of present and hanging out. Incidentally, we may see some of these particles of COVID inside of these kind of cells, or the cells themselves already have viral RNA inside of them and have been enslaved into making more copies of the virus. So our key is to find this guy and to kind of separate it out and know that it's there, and distinguish it from the cells that are actually supposed to be there. So I've made this look like a really kind of easy problem. It's like, okay, well, we've got some cells and we've got some viruses, but there are two problems that our test is going to have to solve. The first problem is the picture can more realistically be shown as this. We have some of the virus, or alternately some cells that have the virus inside of them, but the number of normal cells, especially early on in the case of an infection that's only a few days old, there are a massive number of cells that are in fact supposed to be there. And so we have to kind of find the needle in the haystack, as the old canard goes, where we have a lot of human genetic material and a relatively small amount of viral genetic material. And we have to sort of pluck out that viral genetic material from everything else that we're analyzing from some sort of tissue sample or swab. The other problem that you have to solve is let's consider for a minute a light bulb. So let's say for a minute that this light bulb is dimmable, so it's not just an off-on sort of thing, and let's also say, for sake of argument, that it is plugged in. Can you really tell me at this moment that this light bulb is totally off? Well, no, you can't, especially on this bright white background. There might be a very slight glow here, but we're never going to be able to pick it up, considering that relative to background, there is very little signal. There's very little light. We have basically a kind of a dim bulb, and maybe if we were to darken, everything else around it, we'd see that in fact it was glowing. This is a problem that we scientists tend to refer to as a limit of detection problem, which is to say that this light bulb might be on, but on at so faint a level that you can't actually detect it. So if you have a low amount of genetic material present, or a low amount of COVID genetic material present, that means it's a very small signal, which we might not be able to detect with our sensors, much as you couldn't detect a very faint glow in this filament with our eyes. So what does the test need to do? What does it need to accomplish given these two problems? First, it needs to discriminate between things that belong to the coronavirus and things that belong to humans. And second, it needs to amplify the things that belong to the coronavirus so that we can actually see them, or we can generate a large enough signal that allows us to make a diagnosis. So what is it that we're going to try to detect? If you were going to take a look at one of these macromolecules, which one might you pick? Might you say, okay, this is the one? Well, DNA has some advantages. It's stable. You can extract DNA. It's good for 24 hours. You can freeze it almost indefinitely. You'll still be good. Problem is that there are relatively few copies of DNA present, and more fatally, this is an RNA-based virus. So if you go in and you're looking for coronavirus DNA, you're never going to find it because there is no coronavirus DNA. In RNA, the advantage of RNA is that there are a lot of copies of it, and this means that as this virus is trying to crank out as many versions of itself as it can, it's basically forcing the cell to run a copy machine. And you've got a lot of viral RNA floating around. And of course, it's something that you can detect, and it's something that is present in the coronavirus. The disadvantage of RNA is that it is relatively unstable. In a cell, it has a half-life of about 5 minutes. So whereas DNA is so stable that you can keep it frozen for a very long time, RNA, you generally have, even in a frozen sample, you got about a day to work with it. And if you leave it alone on your bench top for more than about an hour, forget about it. You're not getting anything out of that RNA. So this is actually, there's actually a very good reason for this, it being a communications molecule, or how shall I phrase this? It's actually good for our cells that RNA is short-lived. And if you can think about it, DNA, you want DNA to be very, very stable. Because the DNA that's inside of you, that's sort of, you're hoping that that will stick with you for life. And that's what's, again, the instruction manual, you want that to be relatively immutable. But RNA, let's say that we've made some RNA in our cells. If this were to stick around for a very long time, if the RNA were to stick around for a very long time, protein would continue to be made from that individual strand of RNA. It would continue bumping into ribosomes and being made into protein. So if a cell has some sort of external stress and all of a sudden has to ramp up production of different types of proteins in response to that external stress, if the RNA were stable, it would first have to cancel it, or it'd have to get rid of it somehow, and basically prevent it from being translated. Whereas if it falls apart fairly quickly, you need a fairly continuous signal coming from the DNA to be making a lot of protein in a row. So again, there's an advantage to RNA being unstable in everyday life, but it makes these sorts of detection problems a little bit more tricky. You could also technically go after the protein, which is found inside of your cells and see if there are any viral proteins present. The advantage, again, is that there are relatively a lot of copies of protein. But the disadvantage is it's hard to discriminate between where the protein came from in sort of a testing environment. It's hard to say, okay, this is definitely a COVID protein, this is a normal human protein. It's also very hard to amplify proteins. If you have one protein, you can't make extremely large numbers of that protein. Whereas if you're dealing with either DNA or RNA, you can make a lot of copies of it very quickly, as we're about to see in the next few slides. So the thing you're going to try to detect is RNA. That's kind of the winner. Now, we're in luck because it just so happens that there is a technique to amplify RNA and DNA, and it's something called the polymerase chain reaction. This is a specific type of the polymerase chain reaction called reverse transcription polymerase chain reaction, or RT-PCR. So you have your RNA that you're trying to analyze here, and you add in a protein called reverse transcriptase. Incidentally, this is actually a viral protein. This is the sort of protein that retroviruses use to take RNA, make DNA, and integrate themselves into host cells. So we can make this, we can purify this, and we can, ship it in a series of chemical reagents in order to make DNA. And DNA has the advantage, again, of being stable. So this allows us to further manipulate the DNA. It allows us to make additional copies of the DNA, and we don't have to worry about things just kind of falling apart. So with the DNA, we use something called DNA polymerase, and that is what allows us to make many, many, many copies of DNA. So that's nice. We now have something that we can potentially detect. We should have enough of these DNA molecules that we can say, all right, we either had this viral RNA or we didn't. But how are you going to be selective? And fortunately for us, PCR also allows you to choose what it is that you amplify. So this is how it works. You have two strands of DNA here. You have the sense strand and you have the antisense strand. So these are the two strands of a double helix that we all know and love, and we've seen on, CSI New York and CSI Miami and CSI Las Vegas and NCIS, CSI, that kind of thing. And no, incidentally, we don't sit around looking at double helices all day in our laboratory. We just look at a big long strand of A's, T's, G's, and C's. It's not as glamorous as it looks on TV. Anyway, you have the sense strand, the antisense strand, and you separate them by heating them at 95 degrees centigrade for about 15 seconds. This will break the two strands of your double helix apart. Now, this is where sort of the magic of PCR happens. There's something called an annealing step, where the temperature is lowered to 60 degrees Celsius for about 30 seconds, depends on what protocol you're using. And you have two little pieces of DNA called primers that you've also introduced into this whole sort of batch and mixture. And what's really kind of interesting about PCR is it does not work unless these primers have actually bound either the top or bottom strand. So let's say that they do. One primer binds this top strand, the other primer binds this bottom strand. And I can actually design what these primers look like. I can sit down at my computer and tap out a series of letters, send them out to a company, and I get them back within about 24 hours. And through clever design of these primers, you can get them to bind very selectively to only certain nucleic acid sequences. So if you had, say, an entire organism and you just want to isolate one gene, if you choose your primers so that the primer binds immediately upstream of the gene and immediately downstream of the gene, you can then isolate that gene and use it for sort of further manipulations. It's an extremely powerful tool, and it's very, very commonly used. But in our case, we're also interested in what happens if the primers don't bind. So if the primers do bind, we subject this whole sort of setup to an extension step, which is at 72 degrees Celsius for between 1 to about 5 minutes, depends again on how long the thing is that you're amplifying, in the presence of a DNA polymerase. And what that will do is create a copy. So essentially, the complementary DNA is filled in here by the DNA polymerase, and is also filled in here by the DNA polymerase. So we end up with our primer sort of being integrated into this new double helix up here, and this new double helix up here. So we've taken one double helix and we've made two double helices. We've made two copies of this DNA where there used to be one. All right. Now, if the primers don't bind, if this is not here and this is not here, then this extension step doesn't do anything at all, and you don't actually see any duplication or any amplification of the DNA. This is an entire cycle. So you take all three of these steps and you can repeat them, say 25 times. And if you repeat this 25 times, you make 2 to the 26th copies of each and every one of these double helices, which is over 67 million copies of each individual one. Now, of course, there can be issues. It's never perfect. Probably will make fewer copies than that. But again, you're taking a small amount of DNA and you're making an extremely large amount of DNA in a very short period of time. All right, so this entire process can take between 1 and 3 hours from loading in the DNA to actually running the thing. Right? So we've now made an enormous amount of DNA, but how exactly do we derive a signal from it? Something that we can actually see. Well, there are two ways to do this. One of them is non-specific, and one of them is more specific. So you can take a dye such as DRAC4 or HOST 33342. Somebody needs to tell me how to pronounce HOST, but I think that's about right. And this dye um, is something that, if it is floating freely in space, is not fluorescent. However, when it binds, either when it binds in an intercalating fashion, in other words, when these rings kind of stick into the DNA, I was really looking for a good image, but this is the best I could find. When it sticks into the DNA, then it fluoresces. Um, or similarly, you can have this sort of dye which binds on the face of the DNA. So basically on the face of the double helix. And again, it only fluoresces when it's binding to the face of it. So basically, this is telling you how much DNA you have. If you have a lot of DNA, you're going to have a lot of signal. If you don't have much DNA, you're not going to have much signal at all. So you can imagine if we've done a huge amplification of DNA, of the complementary DNA, which was made from the RNA of the coronavirus, you're going to see some kind of a signal. So again, large amount of DNA is the result of amplification, and using one of these two dyes, you would get a large amount of visible signal. The disadvantage of this, there are two disadvantages of this. First, let's say that your sample prep wasn't very good, and you included a lot of double-stranded DNA, either from the human cells or the other cells that were present, or that you just had some sort of a mistake with the amplification and you amplified the wrong thing. What's going to happen is you're going to see a very strong signal. You're going to see that signal fairly early, so you're going to basically see a lot of noise, but that can be a real big pain. Again, because these dyes don't care where the DNA came from. They're just telling you that there's a lot of DNA present. So that's one problem with them. The other problem is, whenever you have a small molecule that binds DNA, that's kind of scary, because if something is capable of binding DNA, it is capable of messing up that DNA. It's capable of preventing it from being properly replicated, or it might cause errors in the replication of the DNA. So if you were to ingest one of these dyes, and it were to bind to your DNA, it's going to introduce mistakes in the way that that DNA is replicated. And if you have a lot of mistakes in the way your DNA is replicated, you might get cancer. So these dyes tend to be carcinogenic, and you tend to use them very carefully, for that reason. There's another method that's a little bit more specific and perhaps not as scary, where you take these primers that you designed and you functionalize them with a dye, which I've represented as a ball here, basically. And the way that it's set up is that when the primer is ordered, it's in this hairpin shape. So the dye is close to this little piece of DNA, this DNA primer that you have here, and this is switched off. You do not actually see fluorescence from it. This is a process that is known as quenching, where certain dyes brought into the proximity of certain small molecules or certain multimers are not actually fluorescing. I don't want to get into the why and wherefore of that. That's, again, a separate video, but we say that this is quenched. However, when the primer actually binds to the complementary DNA, this hairpin is relieved, which means that the dye sort of springs out and it is no longer quenched, which means that it's fluorescent. It's turned on. You've basically switched on a light bulb. And as you're duplicating it, remember that the primers are being integrated into this new double strand of DNA that you are creating. And you've essentially tagged all of the new copies of the DNA that have your primer on them with this molecular dye. So if you get enough of these guys, so this is a second method. The disadvantage of this method, however, is that you have to functionalize the primer. So this can be a little bit challenging. Whereas here in this method, the sort of non-specific method that we see on the left, you don't actually have to worry about functionalizing your primer. All right. Okay. So there are two kind of big takeaways and two important things as to how this works. First, amplification only happens if the primers bind, which only happens if coronavirus RNA is present. And again, you are able to design primers that are specific enough to recognize just coronavirus RNA in this entire big soup that's happening here. Um, all right. And the reverse transcriptase, I believe, also requires priming. So I think you're only getting the double-stranded DNA that is primed, or that the primer actually attaches to. I'll have to double check that, but that's what I'm thinking, that's what kind of pops into my head at the moment. Even if it doesn't, even if you are getting reverse transcription of all of the RNA that's present, the complementary DNA, or the DNA that's made by this method, the only complementary DNA that is being amplified is the coronavirus complementary DNA. Okay? And if there's no amplification, that means that there is no signal, or there's a signal that's simply too small to actually measure. It's well below the limit of detection. Whereas if you do have amplification, you do have a signal. So you get basically a yes-no response here. So to recap, coronavirus is an RNA virus, and we want to detect its RNA, either floating between cells or inside of cells that are already infected. PCR allows us to amplify specific pieces of RNA or specific pieces of complementary DNA that have been generated from coronavirus RNA. So we are, again, finding that needle in a haystack. If there's no coronavirus RNA present, there's no amplification, and so there's nothing to detect. But if there is coronavirus RNA present, we get amplification and a large enough signal that we can actually measure. So that's how RT-PCR is used to give you a yes-no response with coronavirus. But you can also play some other really interesting tricks with this using a technique called RT-qPCR, or reverse transcriptase quantitative PCR. So inside the human body, and we're not talking about outside sources of RNA at this point, or viral infection, you have things like red blood cells, you have things like nerve cells, you have things like liver cells, you have pancreatic cells. I spent half of my PhD looking at this particular chart, so it's very kind of near and dear to me. And you've got, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 different kinds of cells in just one organ. Not all of these cells occur all of the time. So these are more sort of cells you'd see in an immature pancreas, but the sort of big, I guess, let's see, 1, 2, 3, 4, 5, 6, 7. The big 7 of these that you'd see in a mature pancreas are these cells. And they all do very different things. So the acinar cells produce digestive enzymes. The ductal cells are basically there to not get chewed up by the digestive enzymes. The beta cells are there to regulate insulin, and there are other endocrine functions that are fulfilled by these 4 other endocrine cells. But how do you differentiate among them and say what kind of cells you're actually looking at? Or, perhaps more to the point, let's say that we have some cells that we don't want to have there. We have some cancer cells. How do we tell that the cells are cancerous or that the cells are unhealthy? Well, the question is, how do I tell all of these guys apart? The answer, once again, comes from our central dogma of biology, is that different cells will have different patterns of gene expression, and so there will be different types of RNA that are hanging around. So remember that different cell types make different amounts of protein and different types of protein. So for instance, digestive cells in the pancreas, the acinar cells, produce a protein called amylase. which is used for digesting sugars, whereas beta cells produce insulin. And I think everybody knows what what insulin is, but it doesn't produce any digestive enzymes. So I can tell basically whether I'm looking at an acinar cell or at a beta cell based on what proteins are being produced. And because the types of proteins that are being produced is going to be dependent on what kinds of RNA are present. If you have different cells making different proteins, their RNA is going to be different. So by analyzing what sorts of messenger RNA are present using this kind of PCR technique, I can give you a pretty good estimation as to what kind of cell that actually is. So if you have, in short, more RNA from a certain gene, you need fewer amplification cycles before you actually see a signal. So let's say that you need, let's make up a number, 10 million copies of DNA, again, this complementary DNA, before you see a signal. But before you started the experiment, you had 5 million copies of a particular type of gene. Let's say that it's insulin. You would only need one cycle before you actually saw a signal. Whereas if you had only 1,000 copies of that messenger RNA of insulin, you would need many more cycles before you could actually cross that 10 million copy threshold and actually see a signal. So that's pretty darn cool, isn't it? All right. So you measure how many cycles it takes before you detect some RNA, and you compare that to some kind of reference gene. So there are certain genes that are expressed at roughly the same levels in all of the cells of the body. These tend to be things like DNA polymerases, things that are maintaining sort of order and housekeeping inside of that particular cell. So you say, all right, relative to this reference gene, if I'm seeing a lot of cycles needed to get, say, insulin, there must not be very much insulin present, or rather, if I'm having to amplify any insulin RNA a heck of a lot before I see a signal, that means there's got to be not very much insulin present. All right. So other uses for PCR, again, are things like crime scene investigations. So if we are trying to determine that Colonel Mustard did in fact kill Mr. Body in the kitchen with the revolver, Colonel Mustard is usually not so kind as to leave huge, enormous hunks of DNA lying around on the revolver or at the particular scene of the crime. So what we need to do is amplify the DNA that we do have, which will give us enough to actually analyze and compare to all of the suspects that are in our particular game of Clue. So this is where you see, PCR being most frequently used in television. There's also a really kind of interesting technique called error-prone PCR for something called directed evolution, which won the Nobel Prize for chemistry in 2018. And the basic idea is that proteins are able to evolve. It's not just organisms that evolve, it's proteins that are changing over time. And what happens is, as DNA is being replicated, as genes are being replicated, every so often, the machinery inside of the cell makes a mistake and makes a mutation. And in some cases, or in a lot of cases, this mutation makes no difference whatsoever in the function of the protein. It's a silent mutation. In many cases, that mutation will mess up the function of the protein. That protein is no longer particularly useful. And in a very rare few instances, that mistake is actually beneficial to the function of that particular protein. And if it's beneficial to the function of that protein, it is therefore beneficial to the host organism or to the host cell. That cell then has an evolutionary advantage and is able to sort of predominate. And that's how you get a mutation, a beneficial mutation, sort of becoming a little bit more widely spread. And there you go. So that's evolution from sort of the protein chemist's angle. So the thinking with directed evolution is, what happens if we make a whole bunch of mistakes in copying DNA, in replicating DNA on purpose? We use a DNA polymerase that makes a lot more errors. Then we're going to see more mutations in these proteins. And then what we can do is select the proteins that are actually doing a particular task better. So in other words, we have a whole bunch of random mutations. We select the proteins that are actually more useful to us. And then we can do subsequent rounds of evolution in order to optimize that protein for either the function that we want it to have, or in some cases, for completely new functions that are not found in natural biology. It's a very, very cool field. One of the, the most famous practitioner of it is Frances Arnold. I don't know if she has a YouTube channel or not, but she was the one who won the Nobel in 2018, and we at Caltech were very pleased that that's how that happened. So directed evolution is another use for PCR. So that's about what I wanted to talk about today. So please like, share, and comment. And I'm going to start making physics videos and start, well, actually teaching my class online. So thank you very much, and I will see you next time.